Hi everybody, my name is Tyler Foster, and I have been meaning to do this lecture for quite a while now. Thinking back to medical school, when I was taught the eicosanoid and the cyclooxygenase pathways, it's a little bit confusing at the time. I was hoping to make a video to hopefully clarify the eicosanoid kind of general big picture, then also kind of touch on some clinical applications of this pathway. So let's go ahead and begin. We're going to start out with the general category of eicosanoids. And let me get a different color here. The eicosanoids. Alright, so what are the eicosanoids? It's going to be a 20 carbon structure. So we're going to be talking about kind of a long branch. Remember from organic chemistry, we have those long fatty acid tails. So it's going to be a polyunsaturated fatty acid tail involving 20 carbons, so it would be actually quite longer than this. Now, when I say eicosanoid, what does that mean? Well, it's going to include three general categories of molecules. It's going to be the prostanoids, the leukotrienes, and the lipoxygens. So when I say prostanoid, it can mean a prostacyclin, a prostaglandin, or a thromboxane. So now I'm kind of diving into the deep end right now. I don't mean to do this to confuse you. However, this is going to be kind of the basis of the body. So when somebody says that they're going to be talking about a leukotriene, they're going to be talking about this 20 carbon polyunsaturated fatty acid that's been modified, while if I say a prostanoid, it's also going to be a 20 carbon polyunsaturated fatty acid. However, they're going to have different functions within the body. So prostanoids can be broken down into three different kind of main categories. So remember from biology, where you have kingdom, phylum, domain, all those good things. It's kind of what we're doing here. We're going to start with the main category, and then slowly break down into further topics. So if I say animals, then I break it down into dog, cat, and then dog you can break down into golden retriever, chihuahua. I don't even know how to spell chihuahua. So this is what we're doing. Eicosanoids would be kind of like the general category. We have three different species, so a dog, a cat, and an alligator. And then we can further break down these into more specific molecules such as prostacyclins, prostaglandins, and thromboxanes. Okay, now that I've beaten this into a bush, you can see that we have different prostaglandins, and I'll discuss the functions of each of these within the body. This is going to be a big picture slide. This is probably one of the most important slides that I have in this whole lecture. It kind of gives you an outline of how everything fits together nicely. Now remember I said that you have this 20 carbon fatty acid, polyunsaturated fatty acid. So we're going to get our fatty acid from these little tails here, because remember these are the hydrocarbon tails. We're going to use an enzyme, phospholipase A2, to break those down into arachidonic acid. So we're still taking our 20 carbon structure. Next, we can use two different sets of enzymes. We can use the lipogenase enzymes, or we could use the cyclooxygenase, so the COX-COX1 and COX2 enzymes. These get broken down into different molecules, as you see here. For the most part, this is going to be a complete picture. There are other pathways that I don't have listed, but this is kind of a general outline. So what I would recommend for you to do is grab a piece of paper and copy this down, because if you have this staring at you during this whole lecture, it's going to make things a lot easier. However, if you're not going to take the time out to do that, I also have it kind of broken down into more of a focused picture. So remember, I'm going to flip back here. We have the arachidonic acid here. What I'm going to be doing is I'm just going to be kind of focusing on this part of the pathway. So that's what you see here. We're going to first start out with the leukotrienes. One of the easier topics, if you ask me. Leuko means white, and cyte is cell, so white blood cells. So leuko makes you think, oh, it's going to be found in white blood cells. So leukotrienes are mainly going to be produced in white blood cells. That's a huge point that I want to drive home, because any clues that you can use within the words, it's going to help in this lecture, because everything sounds the same. Eicosanoids, prostaglandin, prostanoid, everything sounds the same. So use the clues that you have available. Now, white blood cells are kind of the immune cells of the body. Leukotrienes are going to be used to create inflammation. So when you stub your toe, your white blood cells will create leukotrienes and inflammation. That's why you get that throbbing feeling. So inflammation can be caused by a whole bunch of different things, including the leukotrienes. And you see here, this is how it's made. So we have Leukotriene A4, C4, D4, E4, and B4. All right, lipogenase enzyme is going to be used to make this. So let's focus on Leukotriene A4. So we have our fatty acid, it gets broken down. We have our 20 carbon fatty acid tail now. We're going to use the lipogenase enzyme to modify it into arachidonic acid 5-hydroxyperoxide. Now, we're going to start out by our first modification to Leukotriene A4 here. It is going to be the gatekeeper, so we can either change it into a B4 or a C4. Let's say that we decide to make it into a B4. We're going to be using the enzyme Leukotriene A4 hydroxylase. And what we're going to be doing is we're going to be producing this molecule that promotes inflammation. Remember I said leukotrienes are made in white blood cells, so Leukotriene B4 can act and promote inflammation. It also allows white blood cells to attach to the vascular endothelium and extravasate. So as the white blood cells kind of rolling along here inside this nice red artery, so we have a blood vessel here. Let me see, let's change color. We've got this white blood cell. He's going to be rolling along. B4 is made, which can act to help him attach to the wall of the vessel, so the endothelium, and extravasate, meaning it leaves the blood vessel and enters some tissue, like let's say it enters the liver here. So the perihepatic tissue, just for example. It also can act as a chemoattractant. So B4 has a couple different abilities. It acts in 1, inflammation, 2, helps the white blood cells extravasate, and 3, acts as a chemoattractant. So let's say the white blood cells now kind of around the liver. It's going to release all this Leukotriene B4. And what that does is it's going to tell other white blood cells, hey, this is where we need to end up. We need to concentrate our attack on this area of the body. Now we also have other options. So our Leukotriene A4 can create the B4, or it can create 3 other molecules. Now in general, again, all of these are going to be involved in inflammation. However, C4, D4, and E4 also can act as a bronchoconstricting agent, a vasoconstricting agent, and a smooth muscle contractant. So just remember, leukotrienes C, D, and E4 are all bad for asthmatics, because what it'll do is it'll bronchoconstrict. It'll close the airway. So if you have any sort of inflammation, that is going to be bad. Here's a little bit of a side note here. I'm kind of teaching this lecture at many different levels, kind of as a basic and then more of as a clinical application. So why do we actually need to know this? Well, we have these drugs in our tool belt called leukotriene receptor antagonists. So that'll be like our montelukast, our Leucast drugs. And montelukast is better known as Singular. So that's a drug that you might have heard on TV. You might actually take it yourself. So how does a leukotriene receptor antagonist work? Well, let's say you have this cell. We're just going to be looking at the very top part of the cell, because this cell is going to be a big cell. On this cell surface, we have a receptor. It's going to be our leukotriene receptor, because we have tons of receptors. This might be an alpha receptor. This one might be a cholinergic receptor, but we also have a leukotriene receptor denoted here. What our leukotriene receptor antagonist is going to do is it's going to come in and put a stop to this receptor. It's going to create a little force field or a block to this receptor alternating the structure of it. So when a leukotriene, so let's say we have a white blood cell here, shrunk way down to proportion. We have a white blood cell that comes along. It's going to activate its lipogenase enzymes. It's going to create our leukotrienes and release a whole bunch of leukotriene, whether it's A4, B4, C, D, E, etc. This is going to eventually try and make its way. So that's what this little triangle stands for, is it's going to be a leukotriene. It's going to try and make its way to the receptor. However, we have our drugs that are going to be antagonists, so they're going to block the receptor so the leukotrienes don't promote inflammation. So we have less inflammation in the body, we have less bronchoconstriction, we have less vasoconstriction, just less response to our white blood cell products. Now, we're going to jump to another topic, which are going to be our prostanoids. So looking back at our big picture, we had two different options. We just covered this route through the lipogenase enzymes. Now we're going to go through the cyclooxygenase pathway, or our COX-1 and COX-2 pathway. So I have it kind of broken down here. We're going to focus on this. Remember, in our very initial slide, we talked about the different naming system. We have prostacyclins, prostaglandins, and thromboxanes. All right. Now, prostacyclin, it's still going to be our 20 carbon molecule. So everything that we're going to be talking about is going to be involving our 20 carbon fatty acid molecule that we're just kind of modifying. So what a prostacyclin is it's going to be specifically prostaglandin I2. So when I say prostaglandin I2, it's interchangeable for the word prostacyclin. So that's an important note here. What it does in the body is it's going to vasodilate, bronchodilate, and it's going to prevent platelet activation, so no platelet plug. So let's think. What we just talked about were the leukotrienes. Leukotrienes, what I told you they did was they bronchoconstricted. They vasoconstricted. So this prostaglandin I2 acts in a different fashion. Instead of being bad for asthmatics, it may have some protective function in asthmatics because it'll bronchodilate. So what we're going to be doing is we're going to be converting prostaglandin H2 into our prostacyclin. All right, so now let's focus on our prostaglandins. Surprise, they are all still 20 carbons. What they're going to be doing is they're going to be used for autocrine and paracrine function, not endocrine. So do you know what the difference between autocrine, paracrine, and endocrine are? Well, as a brief kind of description, autocrine, paracrine, and endocrine. So these are kind of my short notation here. Autocrine is going to be acting upon itself. So let's say we have a cell here, here's a cell here, here's a cell here. This is going to be cell 1, 2, and 3. If cell 1 makes a prostaglandin, so let's say it makes prostaglandin E2, it's going to release it, and that molecule that it just released is going to turn around and act upon itself. It'll release some sort of stimulant to itself saying, hey, we either need to make more or make less. It's going to be an autocrine function. It acts upon itself. Paracrine function means it'll act in the vicinity of the cell that made it. So let's say cell 1 makes some prostaglandin E2 that makes its way over to cell 2, and it's going to act or exert some effect on cell 2. Maybe it makes its way to cell 3. However, it's not going to enter the bloodstream, travel to a remote toe, and act upon some distant structure. It'll stay in the general vicinity of where it's made. So, local area. It's like buying local. Endocrine is when you're going to enter the bloodstream. And it's where you're going to deliver your products. So Cell 1 makes a whole bunch of prostaglandin E2. It'll enter a blood vessel. It'll travel all the way to the brain and exert some function on the brain. So remember, prostaglandins do not function through the endocrine route. So it does not enter the bloodstream, but that's just an example. All right, so what does a prostaglandin do? Well, mainly they're going to be used to induce birth, maintain patent ductus arteriosus, treat peptic ulcers, vasodilates blood vessels, etc., because these kind of go along with vasodilate blood vessels. It'll treat glaucoma as well. So let's focus here. We have our cyclooxygenase 1 and our cyclooxygenase 2 enzymes. So these are kind of two separate enzymes that get lumped together. However, they are going to be two separate enzymes. In a nutshell, the basics of the difference. COX-1 is going to work full time. It's going to be our hard-working enzyme, while COX-2 can be induced. It'll work part-time. Both are going to do the exact same thing. They're going to convert arachidonic acid into prostaglandin H2, which we'll talk about as our gatekeeper. So they're both going to have the same function. They're both going to be found on the endoplasmic reticulum. COX-1 enzyme, remember that's our full-time worker. It is going to be found all over the body. However, it's also going to be working full-time. It's going to be protecting our stomach lining, our mucus lining. So its main function is to be working and pumping out full-time so it can protect our stomach lining. Because remember, the stomach has a pH of, if I remember right, about 2, which is extremely acidic, and it's going to eat through a lot of cells. So we need something to protect our stomach. COX-1 is going to work full-time, and it's going to protect our stomach lining. Now, cyclooxygenase 2 enzyme is going to be inducible. Now, what does inducible mean? Well, it's going to mean that you can induce it or activate it to work harder. Normally, it's just kind of chilling around, playing Xbox. However, you're going to give a signal to the COX-2 enzyme saying, hey, you need to do some work, so it'll get up off the couch, stop playing Xbox, and do some work. And then as soon as his work is done, it'll go back to playing Call of Duty. All right, so that's what inducible means. It means sitting on the couch, playing Xbox until the time comes. Again, it's going to be found all over the body. Now, both of our cyclooxygenase enzymes are going to convert our arachidonic acid into our H2 Hummer. It is going to be our gatekeeper Hummer. That's how I remember it, at least, the H2 Hummer. It's going to act as a precursor to the synthesis of all of our prostacyclins, our prostaglandins, and our thromboxanes. So remember I said prostacyclin simply just means prostaglandin I2, and then we have our prostaglandins and our thromboxanes. All right, so let's talk about prostaglandin D2. It's going to be converted from our gatekeeper H2 Hummer into prostaglandin D2 via the enzyme prostaglandin D2 synthase. Now this is going to be our master prostaglandin. D2 is going to be our master. Now notice how I spelled it. It's not a typo here. I spelled it specific for a reason. It's going to be our master prostaglandin because it's going to be made by mast cells. And if you remember what mast cells do, they're going to be our histamine release cells. So prostaglandin D2 is going to be made by mast cells. And what it's going to be doing is prostaglandin D2 promotes immune cell recruitment. So we're going to have a mast cell release some D2. It's going to promote a whole bunch of immune cells to come and fight to the area. So it'll bring in some white blood cells, etc. It also bronchoconstricts, vasodilates, and possibly does it have some role in reducing body temperature during sleep? Could be. But the main point that I want you to take away is it's going to be produced in mast cells, and it's going to promote immune cell recruitment. Next, we have our prostaglandin E1. I do not have it here on the chart. And why is that? Well, it's because I made this nice, beautiful chart. It fits well together, except E1 is kind of like the oddball. It's like the stepchild. It just doesn't fit into the group. Kind of like Cinderella. It didn't fit in with the stepsisters. So prostaglandin E1 is going to be used because it has an actual drug clinical correlation. Alprostadil is going to be its drug name. And what it's going to be used for is it's going to promote a patent ductus arteriosus. It's going to have some role in helping with erectile dysfunction, helping correct limb ischemia. So what it does, I'm going to back up here, is it's going to vasodilate for the most part. So E1 really doesn't fit well off this chart, but it is made off of prostaglandin H2 to some degree. What it's going to be used for is pretty much keep blood vessels open. So if you have a shunt, what you'll need to do is you'll need to maintain a patent ductus arteriosus as a neonate to provide oxygen to your body. So what it does is you give this PGE1, it's going to vasodilate. It's going to maintain an open artery. Next, if you have erectile dysfunction, what you're going to do is you're going to vasodilate, promote blood flow to a certain region of the body. And then same with limb ischemia. If you have, let's say your foot is not getting the adequate blood flow, you're going to be able to give a drug that's going to vasodilate and promote limb blood flow. Now it's produced from dihomogamma linolenic acid. It does not fit nice, so I did not add it. However, not sure what level you're trying to learn this. However, if you need to know what the function of prostaglandin E1 and the synthesis of it, I provided some basics here. So back to our chart, since it works so nicely, we have prostaglandin synthase. Now, prostaglandin E2 also has a different name. It's going to be called dinoprostone. It's going to have multiple effects. I'm not going to list them all. However, what I do want you to know, its main effect is going to be cervical ripening and uterine contraction. So if you have a woman who is 41.5 weeks into her pregnancy, remember a normal pregnancy is 40 weeks, so she's past her due date and she wants to deliver the child. What you can do is you could give this prostaglandin E2 analog or a prostaglandin E2 drug, and it'll promote cervical ripening and uterine contraction to hopefully induce a pregnancy. Or it could be used as an abortifacient during early trimester pregnancy to abort the child as well. However, it also has tons of other options of use, so it's not limited to pregnancy. It is found naturally within the body. Prostaglandin F2A, it's going to be called dinoprost. What its role is going to be doing is, again, uterine contraction. Does it have some use as a cosmetic? Yeah, I definitely know that there are drugs out there that kind of use this drug to promote eyelash growth. Don't ask me how it got found out that this drug promotes eyelash growth, because I don't know anybody that would want that, but I guess it's popular out there. Thromboxane A2. So we jump down from our prostaglandins and our prostacyclin to now our thromboxane. So thromboxane A2. Again, it's going to be our 20 carbon structure. It's going to be produced by activated platelets using this enzyme thromboxane synthase. So remember, our platelets are going to be used for clot formation. So our platelets create thromboxane A2. It involves the necessary enzymatic software here. It's not going to last long. It has a very short half-life. So once it's made, it'll get degraded pretty quickly by the body. But what it does is it's going to activate more platelets. So we have one platelet here, and it's going to produce a whole bunch of thromboxane A1. It gets degraded quickly, so it has to act quickly. However, let's say we have another platelet coming along, and we have another platelet coming along, and it encounters some thromboxane A2. Those platelets that are just kind of rolling along say, hey, something's not right. I see a lot of thromboxane A2 in the environment. So what it's going to do is it's going to stimulate them to create more thromboxane A2. So there's even more thromboxane A2 created. More platelets are still coming along because remember they're just found in the blood vessels. It encounters all this thromboxane A2. It's going to stimulate more thromboxane A2, and etc., etc. This is an example of a cascade. There are a lot of different cascade signals within the body, such as G proteins, for example. However, thromboxane A2 works in a similar fashion where you have the initial cell creates some thromboxane A2. It'll make even more as it activates more platelets, and etc. It'll make even more when more platelets see it. And what this does is it pretty much promotes platelet aggregation. We're going to create a clot. These platelets will all adhere to each other and create a clot. Also has some role in vasoconstriction because let's say you cut yourself. What are you trying to do? Well, you want to create a scab. You want to create a clot so the blood vessel doesn't bleed anymore. But also it makes sense that you want to vasoconstrict because you want to limit blood flow to that cut so you don't bleed as much. So it has a couple different functions. Thromboxane B2 is going to be kind of an extension of thromboxane A1. Remember I said that thromboxane A1 had a very short half-life. Now, what it's going to be doing is this is going to be the active form. This is going to be the inactive form. So we have thromboxane A2, which is going to produce cell aggregation or platelet aggregation. It's going to be degraded into thromboxane B2, which is going to be inactive. So what's the clinical application of all of this? Well, without getting too in-depth, because this is just a basic introduction lecture, you're going to have different drugs. So we have our NSAIDs, our non-steroidal anti-inflammatories. We have our aspirin drugs. Now what these do, they inhibit our cyclooxygenases. So let's take a look over here. This is just an extension of that big chart that I had you draw out originally. So we have our triglycerides, or our phospholipids here. We have our carbon tails, hydrocarbon tails. They get broken down into arachidonic acid, and then they get further processed by our cyclooxygenases. Then we also have our lipoxygenase pathway over here. Our NSAIDs and aspirins are going to block our cyclooxygenase 1 and 2 enzymes. So we're going to not produce as much prostaglandin. Also, corticosteroids play into the picture here because steroids are going to inhibit the phospholipase A2. So both NSAIDs, aspirin, and steroids are going to have some degree of preventing prostaglandin, prostacyclin, thromboxane production. However, corticosteroids are just going to act in a different location. They're going to prevent arachidonic acid from even being made. So you may have different leukotriene or reduced leukotriene production. Remember I said white blood cells produce the leukotrienes? Well, if you know anything about steroids, steroids decrease your immune function. So it's going to decrease all of your immune function through inflammation through our leukotriene pathway. So corticosteroids decrease the amount of arachidonic acid that can be converted into our leukotrienes, which is going to result in a decrease in inflammation through our leukotrienes. Perfect. So different mechanism of these drugs, same ultimate overall idea, decreased prostaglandin, leukotriene, etc. release. That's all I got for you. If you have any questions, be sure to ask. Otherwise, I appreciate any comments or feedback that you may have. Thanks.